This is a continuation of chapter 3. We're going to start with the cytoplasm of the cell. So this is cellular biology part 3, really. So inside the cytoplasm, inside the cell membrane, all the contents of the cell are the cytoplasm and the nucleus. The cytoplasm is the stuff that's between the plasma membrane and the nucleus. Cytosol is the solvent that everything is dissolved in or is suspended in. It really is a complex mixture. It's water with a bunch of solutes, things that are dissolved in it, with inclusions, with there's organelles, and so it's a mixture. Some of the mixture is colloidal, some of the mixture is solution, and some of the mixture is just suspension. So it is complex. The cytosol is considered the solvent. The organelles are structures within the cell that are basically machinery, and inclusions are granules of stuff that are found in the cell, that are visible in the cell. Basically, if you were to break this down and look at some sort of an analogy, so let's talk about a wood stove in a room. The cytosol is the air in the room, the organelle is the wood stove, and the inclusions is the firewood. That basically sums it up. So there are various organelles. Here's a list. It's not a absolute complete list, but it's a pretty comprehensive list. So the organelles can be found as membranous organelles or non-membranous organelles. The membranous organelles are made up of lipid bilayers, much like the plasma membrane. Excuse me. And they include mitochondria, peroxomes, lysosomes, endoplasmic reticulum, and Golgi apparatus. The non-membranous ones are made out of filaments. And so it's the cytoskeleton, the centrioles, or the ribosomes. And we'll talk about the important ones here. So we'll start with the mitochondria. Mitochondria is considered the powerhouse of the cell. Really what it is it provides the site of Krebs cycle and oxidative phosphorylation, the electron transport chain. So without mitochondria, the most the cell can do is make 2 ATPs out of every glucose molecule. With mitochondria, they get 36 ATPs out of a glucose. So most of the cell's ATP comes from the mitochondria. Now, ATP is the charged battery of the cell, as it were. It is where the energy gets stored. It's like your cell phone battery. And so mitochondria really are the chargers for that. Now there is a very good theory that mitochondria were once free living organisms that have invaded our cells and symbiotically live there. They have their own DNA and their own RNA, and it is different from the rest of the cell's DNA and RNA. So maybe they started life as some sort of a bacteria, and they pay their rent by providing a site for ATP production. I'm not going to ask you to know what all the bits are, where the mitochondrial DNA are, and the cristae and the matrix and all of that. Suffice it that it is an enzyme-covered membranous organelle, and those enzymes are used to facilitate all of the chemical reactions in Krebs cycle and the electron transport chain. And so therefore that's the only place that it can happen. These are probably the most important organelles, and when you look at them, or you look at a cell and you see a lot of these, it means that it's a big energy user. If you look at cardiac muscle cells from the heart, you're going to see a lot of mitochondria. If you look at skin cells, you're going to see relatively few mitochondria. And it's just as a reflection of usage by the cells, by the principle of complementarity. Ribosomes are probably the next most important organelle for you to understand. Ribosomes really, I don't care what they look like or what their structure is. They are the site of protein synthesis. So they have some ribosomal RNA, and when we talk about protein synthesis in the next couple of videos, it will make sense. But this is where it happens. This is where proteins get synthesized. Now there's two types of ribosomes. There are ribosomes that are floating around in the cytosol, that are found freely in the cytoplasm, and we call those free ribosomes. And they synthesize proteins that dissolve in the cytosol. There are other ribosomes that are attached to the endoplasmic reticulum, and we call those membrane-bound ribosomes. And they make the proteins that are destined to be incorporated into the cellular membrane, be intrinsic proteins, or proteins that are going to be exported from the cell. So in the case of I don't know, glandular cells, where the cell is making a lot of things to be exported, you're going to see a lot of rough ER, a lot of ribosomes bound to endoplasmic reticulum. And so the all endocrine cells seem to have this. Goblet cells that produce mucus, they produce the protein mucin, which then becomes mucus, would have a lot of membrane-bound ribosomes. The endoplasmic reticulum is this really assembly line of membranes. It's interconnecting tubes, and basically, and it continues to the nuclear membrane. What basically their job is to do is to bring together the components that are necessary to synthesize proteins or to have chemical reactions. So these provide kind of narrow pathways that sequester these materials from being all over the cytoplasm and therefore more difficult to access. So that basically it brings the amino acids with their attached transfer RNAs to the sites of the ribosomes, and it just provides kind of a constant source. The real analogy of the endoplasmic reticulum is an assembly line. It is about keeping everything arriving in order at the right time in the right place so that assembly can happen. So the rough ER is the ones with the ribosome studded on it, and you'll see that it's continuous with the nuclear envelope. We'll see why in a minute, but you'll notice the nuclear envelope has little pores through it, here and here and here. All these little pores open up into this endoplasmic reticulum, and this is where these proteins get synthesized. In the smooth ER, other chemical reactions happen. So, so any protein that's going to be secreted or is non-soluble will probably be made on rough ER. The smooth ER really has enzymes on its surface, and they're involved with cell functioning, with chemical reactions that happen in the cells. So the smooth ER in hepatocytes in the liver help break down glycogen, detoxify drugs, etc. Steroid-based hormones are made here. So if you look at the cortex of the adrenal gland, you will see those cells have a lot of smooth ER. They're involved with transportation and things like that. Smooth ER in muscle cells, cardiac muscle cells, and skeletal muscle cells have a special name. It's called sarcoplasmic reticulum, and it's used basically as a storage bag for calcium. And when we talk about muscles, we'll see that. So if this series of tubes here is the endoplasmic reticulum, and that's an assembly line, then the Golgi apparatus, which looks remarkably similar, it looks like this, is really the warehouse. It's shipping, warehousing and shipping of these proteins. Basically, it is the same sort of folded membranes as the endoplasmic reticulum, and reticulum, by the way, ret and net are the same word, so it really means network. The Golgi apparatus is remarkably similar, except what it ends up doing is it concentrates and packages up these proteins and fats and makes the transport vesicles that will move the stuff to where it needs to go. So we see Golgi apparatus in cells that are going to be exporting things. For instance, in neurons that are going to be exporting neurotransmitters, you will see a fair amount of Golgi apparatus, because that's how a nerve functions. That's what it does. It releases neurotransmitters. The rest of them are fairly minor as far as physiology goes. If we were going to be a cell biologist, we'd be spending lots of time on this. But basically, lysosomes are used. They lyse. They break things up. So some means body. Lysosome means breaking up bodies. And they're basically little bags of digestive enzymes. And their job is to digest any ingested bacteria or viruses or toxins. They break down old and non-functioning organelles. They break down glycogen. They break down bone to release calcium. They destroy injured cells or cells that are no longer useful. We get rid of them. And all of this happens via cells that contain a lot of lysosomes. So macrophages, for instance, which are specialized cells that ingest things, have lysosomes that digest whatever the macrophage ingests. In the liver and in muscle cells is where lysosomes are used to break down and release glycogen. Osteoclasts are a special type of phagocyte that eats bone and releases calcium. So lysosomes are organelles within specific cells. Cells that don't take anything into themselves have relatively few lysosomes. They just have enough lysosomes to maintain their own house, like to get rid of any organelles that are no longer functioning. Cells such as osteoclasts, whose only function is to break down bone and release calcium, have a huge number of lysosomes. And the enzymes that they have are appropriate to the job that is being asked of it. So this endomembrane is really more of the endoplasmic reticulum, Golgi apparatus stuff. So, and with all the little vesicles. So proteins get made here, they get concentrated and stored here, the export vesicle is made out of the Golgi apparatus, and then it floats to the surface and stuff is released. Lysosomes are also made here, and they float around as little bags of digestive enzymes. Perioxomes are the same thing, except what they have is peroxide. So they really are neutralizing free radicals. They help detoxify substances that end up in the cell. The cytoskeleton is really a bunch of microtubules, microfilaments and intermediate filaments that give the cell a specific shape. It's probably useful to think of microtubules to be like little pipes and tent poles, as it were. The microfilaments would be like the threads in the canvas of your tent, and the intermediate filaments would be like the ropes that you use to pitch your tent. A tent needs all three of those things to give it its shape. Microtubules, the tent poles, are resistant to compressive forces. The microfilaments and intermediate filaments are resistant to tensile forces, so being pulled apart. So microfilaments are involved with cell motility, etc., and they kind of give form to it. So the microfilaments look like this, and you can see that this cell really has the form of these blue microfilaments. In certain cells, like muscle cells, they have a much less random pattern. Intermediate filaments are really rope-like protein fibers, so they are for resisting pulling forces, tensile forces, and they attach to places on the cell membrane called desmosomes. And they look like this, and so it looks like ropes. This is a photograph of staining of the intermediate filaments, and you can see that it ends up pulling on things and gives tension across the cell. Microtubules are hollow tubes, and so they're like little pipes. They really look like tent poles, like this. And they are resistant to any compressive forces. So they're like little rods. And so the interaction of these three things is what gives the cell its shape. Other than that, it would just be spherical, just be a bubble. We'll talk about the motor molecules when we talk about muscular contraction. It's a much more appropriate time to talk about it. But it basically looks like this. Centrosomes are really microtubules that are involved in mitosis. So cells that are amitotic, like red blood cells or neurons, don't have these things. And I'm not going to get into it, except that they reproduce, and as part of cytokinesis, and you find them in the daughter cells. They end up looking like this. They're kind of almost like little blinches that are used during anaphase and telophase of the cell life cycle. There are other things that stick out of the cell, cilia and flagella. The difference between cilia and flagella in mammalian bodies, in the human body, is that cilia move crap past the cell, flagella move the cell through crap. So flagella make the cell swim. The only example of it in the human body is sperm cells. Cilia stay put, and the little arms wave back and forth, and they pull substances across the surface of the cell. So basically, an example of, or an analogy of how cilia work is crowd surfing. How you know, in the mosh pit, you end up on everybody's outstretched hands and get passed along at a concert. Cilia is used to move mucus, for instance, in the lungs. Cilia are used to move mucus in your nose back towards your throat. When the cilia stop working, that's when your nose runs. So this is some diagrams of flagella. Cilia is more like this. I don't really care about the phases of ciliary motion. The important concept is that it is crowd surfing. Microvilli are extensions off the cell that increase the surface area. So anytime we see microvilli, the cell is actively absorbing it. So it's a specialized thing we see in cells that absorb from the outside. So it looks like this. It looks like Bart Simpson's hair. Okay, in the center is the nucleus. Now the nucleus is the library. This is where the genetic information is found. And the genetic information is really instructions for making proteins. And all cells do, ultimately, is make proteins. They either make proteins that are enzymes that catalyze cellular reactions, or they make proteins that are structural, or they make proteins that are functional, etc. Ultimately, that's all that it is. So the nucleus is a series of basically books with each gene on each chromosome being a recipe for a specific protein. Most cells only have one nucleus. Some cells don't have any nucleus, and other cells are multinucleate. They have many nuclei. When you think about the size of a skeletal muscle cell, some of them can be almost a meter long. You really need to be able to synthesize proteins up and down the whole length of it. So there's multiple nuclei. In normal small cells, you only really need one. Red blood cells, once they finish making the hemoglobin, have no need for a nucleus anymore. Now, it's one of the reasons that red blood cells can't repair themselves. They can't make any new proteins. They don't have the genetic library for it. They don't know how. There's no instructions. In the center of the nucleus, so the nucleus is surrounded by a lipid bilayer called the nuclear envelope, and that has these little channels called the nuclear pores, and those nuclear pores are continuous with the endoplasmic reticulum. In the center of it is something called the nucleolus, and the nucleolus is the site of ribosome synthesis. It's where ribosomes get made. And when you consider that ribosomes are the site of protein synthesis, and I've just said that protein synthesis is the real job of every cell, then it's pretty freaking important. The rest of the nucleus holds this stuff called chromatin. So the nuclear envelope I just talked about, it looks like that under electron microscopes. Nucleoli makes ribosome subunit assembly. It's involved in making ribosomes. This is probably the most important things. Chromatin are really threads of DNA and the supporting proteins, a little bit of RNA. And chromatin is basically unraveled chromosomes. If you go buy thread at Beauclair or someplace, you're going to buy it on a spool. The thread's going to be all wound up and safely tucked onto a spool so that you can put it in your pocket and go home with it and not have it tangled or become a mess. When you're moving it around, you need it on the spool. When you're sewing with it, you need it off the spool. You need to take it off the spool to use it. Well, it's the same thing with chromatin and chromosomes. Chromosomes are the DNA wrapped up on a spool, which is useful for transporting it without it getting tangled or broken or messed up. Chromatin is the same DNA strands unraveled and off the spool. It ends up looking like this, chromosomes out to chromatin. We'll talk about how this gets duplicated, and we'll talk about how it gets used in the next video.